All right, ladies, so let's talk about water potential. Water potential is important in determining the direction of the movement of water in and out of a cell. It's important not only for plant cells, like we talked about with our Osmosis and Diffusion Lab, but also for animal cells. But for right now, for today, I'm going to be talking to you about the plant cell. And one of the reasons that we talk about the plant cell is that it actually is a little bit more complicated than the animal cell. Just as a reminder, in an animal cells don't have cell walls, and so therefore, if an animal cell is in a hypotonic solution, then that animal cell will probably lyse, killing the cell. But in a plant cell, the normal condition is in a hypotonic solution, and it is when the plant is turgid, and that's the situation the plants like to be in. When the plant cells are flaccid, you would see a plant that's wilted, shriveled up a little bit, looks like it needs a little help, maybe some water, and that would do the trick. It perks right back up again as soon as it hits that water, because water will move back into the cell, adding this turgor pressure, which is the pressure placed on the cell wall by the cytoplasm as more water moves in. And in a plant cell, when it is placed in a hypertonic solution, meaning that that solution has a lot of solute, water will go out of the cell to dilute the solute on the outside of the cell, and therefore the cell becomes plasmolyzed, or what we call it crenellates. And usually that leads to cell death in a plant cell. So going back to water potential and why it's important. It's important obviously for what I just said, homeostasis of water in a plant cell or an animal cell. It's also important in transpiration. And we're going to be talking about this again later in the year. So because of that, you should be thinking to yourself, this is really important. So just to review a little bit, we discussed this in class, but water flows from regions of higher water potential to regions of lower water potential. There are two factors in water potential. One is the solute concentration, which we represent as psi S, and the pressure potential, which we represent as psi P. The equation for water potential, therefore, is psi equals psi P plus psi S. And we're going to go through how this all works in a second. All right, so what is solute potential and what is pressure potential? Solute potential is really a matter of the concentration of that solute in a solution, and that solution could be on the outside of the cell or it could be on the inside of the cell. Solute potential really can be reflected in either case. It really is the concentration of a solution. Now, as you add solute to a solution, that solute potential becomes more negative. Also, I want to remind you that solute potential is sometimes called osmotic potential. And so just so that you know that and just kind of keep that in mind in terms of terminology. All right, a negative solute potential reduces the water potential from the equation from before. As you can see there, that's what would happen. The other thing that I want to mention too is that the solute potential can never be positive. It can never be positive. It's either 0 or negative. 0 would be pure water, distilled water, where there's no solute present. As you add solute, you are making that solution more negative in terms of the solute potential. Okay, pressure potential is the physical pressure on a solution. In a beaker that is open to the atmosphere, the pressure potential is 0. But when we talk about the pressure potential inside of a cell, we are not now in an open system. We are much more in a closed system, and that's where you might see some pressure potential situations. Turgor pressure, like I said before, is the force caused by the cell membrane against the cell wall. And pressure potential, unlike solute potential, can be positive, 0, or negative. Okay, I want us to think about pressure potential a little bit in terms of a balloon. If I have a balloon and it's blown up and sealed on the end, and I squeeze one end of that balloon, what happens to that balloon? Well, the pressure causes that balloon to sort of bulge on one end. When that happens, the air on the inside is moving away from my force of my hand. In that situation, if we're talking about pressure potential, that would have a positive value, okay? Because it's sort of pushing away, all right? It's a pushing situation. Versus, I want you to now think about a syringe. And when you pull liquid up into the syringe, that is becoming a negative value because you're pulling, not pushing, but pulling that solution up towards you, not away from you. And so that would be a negative pressure potential value. It's important that we think about this for later when we talk about trigger pressure and how this relates to. All right, so let's talk about solute potential and solute potential's equation. On the AP Bio exam and on my tests, you will be given both the equation for water potential and the equation for solute potential, along with the R and the T. You just need to know how to utilize this equation. So let's talk about it for a second. The ionization constant, what does that actually mean? Well, it means in an aqueous solution, does that solute form ions? So, for example, if we take a look at, all right, so if we take a look at something like sodium chloride. In an aqueous solution, sodium chloride forms two ions, sodium ions and chloride ions. So the ionization constant for sodium chloride would be 2. We have been using in our lab sucrose. You need to be asking yourself at this moment, does sucrose form ions? And your immediate answer, because you are so good at chemistry, should be no. It is a molecule, it's not an ionic compound. And so therefore, for sucrose, the I is 1. Okay? So it does not form ions, and so its I, or ionization constant, is 1. C is the concentration of solute, and in this case, we represent this in molarity, and molarity is moles per liter. All right, and so that's basically your concentration of your solute. R is the pressure constant. Oh, sorry for that parentheses, but it's the pressure constant, and it will be given to you on any on our equation sheet. But it's 0.083 liter bars over moles per moles K. And you should start to see how liters might cancel, moles might cancel. All right, and temperature, that is in kelvins always, and so it's 273 plus your degrees Celsius. And you can see now sort of how kelvins might cancel out as well. All right. All right, so you should be able to figure out the solute potential for your solutions. If I was to give you any solution concentration, you should be able to figure out the solute potential. All right, so in our experiment in our lab, we made in class a rough graph that looks similar to this. This doesn't look so rough, it looks very professional. Ours was a little rougher. And I said to you, well, we can figure out sort of the situation where our cell would be at dynamic equilibrium. And that was at 0, when you had 0% change in mass, meaning that there was no change from the initial mass of that potato core to the 2 days later. And so that means that water is at dynamic, or that cell is at dynamic equilibrium with the surrounding solution. What we can do then, if you see a graph like this, either on my test or on the AP test, is you need to start thinking about a linear regression line that might look similar to this. And you take that line and you find where it would be at dynamic equilibrium. You go down and you might be able to find out a little bit of something, like the actual concentration of this inside of that cell. Why? Because at dynamic equilibrium, the inside and the outside have the same concentration. And so let's make this 0.33 molars for our cells. All right, let's see what that. All right, so what that means is if I was to calculate the solute potential, remember it's a negative value, I CRT. And you would get negative, remember it's 1, 0.33 times 0.083, oh no, times, I'm going to write this like this, all right, times, let's say we have 293 kelvins. Okay, when we do this, we get a negative 8 bars. Okay, and that would be our solute potential. Excellent. Very nice. So do you see how you can use that graph and that information to figure out the solute potential of a cell that is in dynamic equilibrium? You can figure out the solute potential inside of the cell. That becomes really important. I also want you to think about this for a second in terms of what this graph represents. We've got a linear regression line, a best fit line, and I want you to think about what it means to be on this side of the line and what it means to be on that side of the line. I'm not going to tell you. I just want you to think about that, and we will hopefully you'll think about that, and you might mention that a little bit in your lab write-up. All right, so what does this all mean? Here I've got a picture of a cell that is in a beaker. It's a very big cell if we were thinking about scale. And it's in distilled water, so it's in pure water. All right, because it's in pure water, it means that it has a solute potential of 0, okay? And it also has a pressure potential of 0 because it's an open system, and an open system like this, like a beaker or something, it has a pressure potential of 0, 0 just sort of being a standard, sort of like a regular constant pressure. And so the water potential is going to be 0 in this case for our pure water. So now I want us to think about this cell and the sort of calculation that we did before. I want you to consider it with this cell, and we'll just keep our concentrations, solute concentrations in related to this particular example. So now let's talk about, well, what's going on in here? All right, on the inside of that cell, we have a solute potential of 8 bars from our previous calculation. And I want us to think now about what's happening with that pressure potential. Remind yourself when I talked about that balloon and how that balloon was a positive value for pressure potential when it's forcing the water away. And that's what's happening to the cell wall when water is being pushed on that cell wall from the inside. So actually, it has a pressure potential of 8 bars. And that would equal a water potential of 0. So in this case, water is at dynamic equilibrium. Our two water potentials out are equal, is equal to the one that's in, and that hopefully that helps.